Hi, my name is Sophia Salazar, and I am a rising senior at a high school in the Greater Toronto Area. And my mentor is Rajni, who is a PhD student at the University of Calgary. And she assisted me in studying the TCA cycle, metabolism, and red and white blood cells using isotope tracing with a liquid chromatography mass spectrometer. So today I will be speaking about my research and my experience in the program through these subtopics: introduction and purpose, process, key studies, and reflection. I will start off with an introduction to what my research is about and its purpose. So my whole research project revolves around the idea of metabolism, since I will be examining the different metabolites in red and white blood cells. So metabolism is important because it serves two important and main functions. It provides energy for cellular processes such as growth, and intercellularly enables synthesis and assimilation of new materials. Specifically in the study of metabolism, I investigated the TCA cycle, or the tricarboxylic acid cycle. And the TCA cycle is a series of reactions in a closed loop that creates a metabolic engine within cells. The purpose of this cycle is to synthesize ATP or energy and produce a precursor for other pathways that promotes growth in the cells. So there is an oxidative and a reductive TCA cycle. The oxidative TCA cycle occurs in all living organisms where the cycle goes in a clockwise direction and takes place in the matrix of the mitochondria, while the reductive TCA cycle also occurs in nature, but it has reverse cycling. And the reverse cycling is possible because most enzymes in the TCA cycle catalyze reversible reactions, meaning that they work in both directions. So to examine the study in metabolism, there are certain tools used. And in my research, I use isotope tracing, liquid chromatography, and a mass spectrometer. So isotope tracing consists of unveiling which metabolic pathways are active and inactive, and it also enables an understanding of a metabolic pathway's flux. And flux is the ability of one metabolite to convert to another, which means that when a metabolite has a higher flux, it has a higher and faster labeling rate. So liquid chromatography and mass spectrometer work together because they help analyze metabolism and metabolic pathways. So specifically, liquid chromatography extracts compounds from the mixture and injects these pure compounds into a column. And when the pure compound exits the column, it records its retention time, while the mass spectrometer evaluates these pure compounds based on its mass to charge ratio. So to further explain my research, I wanted to show an image of an oxidative TCA cycle, which again takes place in the matrix of the mitochondria. And the metabolites of interest that I am analyzing in my research, which is present in the TCA cycle, is glutamine, glutamate, alpha-ketoglutarate, citrate, aspartate, malate, fumarate, and succinate. So in this study, I use carbon-13 to measure the flow of carbon. Since carbon-13 glutamine metabolism will also have carbon-13 in them, and this can be detected even with a metabolite that has an endogenous carbon-12. And this flow of carbon is truly determined by comparing the metabolic production behavior of the reductive or oxidative TCA cycle. And this is done through counting the number of carbon-12 and carbon-13 in each metabolite to deduce where the carbon-13 is coming from, whether a carbon dioxide was fixated, how the cycle is moving, and what enzyme was used to create a specific reaction. And the uniformly labeled carbon-13 glutamine is fed into either red or white blood cells to examine the flow of carbon. So you can see here the uniformly labeled carbon-13 glutamine enters the cell, and then it converts to glutamate, alpha-ketoglutarate, etc. So now I'm going to talk about the purpose of my research and why I chose it. So my research really explores the importance of metabolomics technology like the liquid chromatography mass spectrometer or the LCMS. And this is really interesting and important because technology like the LCMS helps further technology and metabolism as it analyzes the activity of metabolic pathways and how the particles like carbon move through these pathways. And it also promotes more advanced technology and more discoveries in biology and medicine. It was also interesting to explore that a mitochondria-free cell, like the red blood cell, does have a flux in the TCA cycle that is reliant on using the mitochondria to function. So it was interesting to see that it's expected that there would be no TCA cycle that was present in the red blood cells to generate energy, but through the results, we do see it. Another reason why I chose this topic is because isotope tracing does have a greater impact in medicine as metabolites can be identified and studied to be linked to human diseases, cancer, and diabetes. So it's really interesting to see how subjects like biology really do link and have a greater impact on the pursuit of medicine. So now I will speak on the process or methodology to extract all of my data. So the sample preparation was done by my mentor at the University of Calgary, while I analyzed the data with my mentor for my paper. So now I'm going to talk about the sample preparation, which again was done by Rajni. And red blood cells and white blood cells, the way that they are prepared are different. So I will explain how to do both. So I'll start off with red blood cells. So first, fresh blood is collected. Then the cells are centrifuged, and this helps remove the plasma and buffy coat layer. Then the red blood cells are washed 3 times with the RPMI medium and the fully labeled carbon-13 glutamine, and then it is resuspended to obtain a 5% final concentration. And once the 5% red blood cell solution is obtained, it had approximately 100 million red blood cells per milliliter. Then it is incubated with fully labeled carbon-13 glutamine for 48 hours. So now I'm going to talk about the white blood cells and how that's prepared, since again, like I said, it is prepared differently. And how it differs is that white blood cells need to be purified. So I'll explain how that purification process goes. So first, the blood is diluted to 50% and it's layered on top of a histopack. Then the white blood cells are centrifuged for isolation, and this is based on the density gradient solution. And as you can see in the image, the white blood cell is between the plasma and the histopack, and it's really small, and it's emphasized by this white circle over here, a red circle over here. And then you can see that the red blood cells are synced to the bottom, and the white blood cells are carefully retrieved from the histopack tube and washed with the RPMI medium twice. And after the purification process, the white blood cells are incubated with carbon-13 glutamine for 48 hours. So after the 48 hours, the samples are extracted from the incubator, and intercellular and extracellular metabolites are removed with 50% methanol. And if we go back over here, the metabolites go through the LCMS using the reverse phase ion pairing method, and this is used to analyze negatively charged compounds like the TCA metabolites, and they're negative because they carry 1 or more carboxylic groups. Now I'm going to talk about how after the data is generated and how it is analyzed through the softwares Maven, which is in step 3, and Microsoft Excel, which is in step 4. So the software Maven was used with the provided data to examine the TCA metabolites for isotope labeling patterns. And in the image here, I selected the metabolite malate. And you can see here there's a graph with peaks, and the data is down here. So then this data was exported into Microsoft Excel, and this software was used to create bar plots for intracellular and extracellular metabolites isotopomers. And you can see this is an example of the graph for malate extracellular. And these plots really help to trace the isotope labeling patterns as well. Now I'm going to talk about the key studies, and I'll start off with the white blood cells. So before I actually start with the white blood cells, I want to talk about the diagram on the right. So you can see here that this is the TCA cycle that was shown in previous slides, and the bar plots are surrounding this cycle. And this shows the carbon-13 isotope tracing pattern in TCA metabolites examined in white blood cells. And this is fed with, again, uniformly labeled carbon-13 glutamine. And in this graph, you can see that the x-axis represents isotopomers, which are isomers with the same chemical arrangement, but different isotope forms. And the y-axis represents the signal intensity. So I'm going to just explain the how to analyze the data and how I came to my results. So the uniformly labeled carbon-13 glutamine is called +5 glutamine isotopomer, as it has 5 carbon-13 carbons, and this gives it 5 mass units heavier than the regular carbon-12 glutamine. So similarly, +1, +2, +3, +4, +5 isotopomer forms of any TCA metabolite indicates that these isotopomeric forms are 1, 2, 3, 4, 5 mass units heavier than the corresponding carbon-12 metabolite. So for white blood cells specifically, I predicted that it will have an oxidative TCA cycle, as white blood cells do have a mitochondria in it, and TCA cycles do rely on the matrix of the mitochondria to function. So that's why I predicted it would be an oxidative TCA cycle. And through the data that is shown in this graph, you can see that it is undergoing an oxidative TCA cycle. And one part that makes this evident is +5 glutamine, +5 glutamate, and +5 alpha ketoglutarate. And this shows that it's oxidative. It's an oxidative TCA cycle because the uniformly labeled carbon-13 glutamine is converting into glutamate and alpha ketoglutarate through enzymatic steps. Then I want you to pay attention to the alpha ketoglutarate to succinate, because it goes from +5 alpha ketoglutarate to plus citrate, and this is done because a loss of carbon-13, since a molecule of carbon dioxide was released into the environment. And that again shows that oxidative TCA cycle is undergone through this cell. Then again, it's sort of predicted that there's a +4 succinate from this enzymatic step, then +4 fumarate, +4 malate, +4 aspartate. Now, what's interesting is the enzymatic step between +4 aspartate metabolizing the +4 citrate, but the +4 citrate is not seen in the data. And one of the reasons why is because of the flux in this enzymatic step is very high, which means that this metabolite is more faster consumed than produced. So I also want to note that the TCA metabolism is cyclic, which means that the carbon-13 labeled metabolites in this second round is also evident in the data, and this is seen by the second highest bar in each of the plots. Now I want to talk about red blood cells, and to note, red blood cells is a mitochondria-free cell, but the data showed that a TCA cycle did occur, which suggests a reductive TCA cycle. Since it's predicted that since a red blood cell does not have a mitochondria, there might not be any TCA cycle that is present, but there was one, which suggests a reductive TCA cycle. So as predicted, there is +5 glutamine, +5 glutamate, +5 alpha ketoglutarate. Now what's interesting and what really proves that this is a reductive TCA cycle is the +5 citrate, because this is only created by the TCA cycle, and this is done because of the isocitrate dehydrogenase enzyme, and this is only done through the reductive TCA cycle, and it basically attaches carbon dioxide to +5 alpha ketoglutarate to produce that +5 citrate, and this is seen through the diagram over here. And another thing to note is aspartate is used as a proxy of oxalacetate, which is OAA here, but the OAA is not seen because this metabolite is very short-lived, which means that there are no results that are seen in the data, which means that the +3 aspartate indicates that +5 citrate loses 2 carbon-13 carbons in the form of acetyl-CoA to produce +3 aspartate. So the reason why this happens is because the presence of citrate lyase enzyme that splits 6 carbon citrate into 4 carbon OAA and 2 carbon acetylene attached to coenzyme A. And the lack of mitochondria means that there's no citrate synthase, which joins acetyl-CoA and OAA together. So the regular conversion between aspartate to malate, the fumarates are all +3, but what's interesting is the conversion between fumarate to succinate, because you expect a +3 succinate, but it is not present. So this suggests that the reductive TCA cycle does not go through a full TCA cycle, since fumarate does not convert into succinate. This further suggests that there is a bifurcation or a breaching point in glutamine. And this is done because of the entry point of alpha ketoglutarate. So it's either the +5 glutarate at alpha ketoglutarate adds a carbon dioxide to make the +5 citrate, which continues the reductive TCA cycle, or losing a carbon dioxide molecule to make +4 succinate, and that would continue the oxidative TCA cycle. So since I've talked about my research, I now want to reflect on my experience in the research program. So first I want to talk about my mentor, Rajni. She was a very knowledgeable and supportive mentor who really helped me throughout my time in the program. She knew that this was an advanced concept for me, and she really helped me through it and gave me a full understanding of this topic. She also gave me many opportunities to learn advanced concepts in areas of knowledge that I was interested in, which is, again, metabolism and molecular biology. And she really, again, helped me fully understand such an advanced concept and kind of enhance my interest in this field of biology. Another thing that she did was expand my learning with past research articles, like the ones that she did with other colleagues at the University of Calgary, or just resources that she found online, or some lecture slides that really helped me, again, further understand my research and become increasingly interested in my research. Lastly, my mentor really helped me have insight on pursuing research in the future, because I had such a positive experience working with her and doing my research. So it really inspired me to further do research in the future and maybe pursue it as a career. The last thing I want to talk about are some takeaways from this experience. I really developed confidence with my academic future and career. Since I had such a positive experience with research, this is something that I maybe want to consider in the future, or just do future research projects, because I really did enjoy studying a topic I was really passionate about and communicating it at the end. I also gained experience in examining scientific data with the assistance of a mentor. It was my first time using the LMaven software, and this is a software that is used in metabolism, in the study of metabolism a lot. So it was really interesting to be able to know how to use such an important software, and it really, again, enhanced my learning and interest in metabolism and molecular biology. It also really helped strengthen my skills like time management, scientific communication, and organization. Since this program has a lot of deadlines and you need to stay organized with deadlines and the data and learning that you have throughout the program. This really helped me strengthen these skills and will really help me in the future. Another thing that this program did for me is help with my scientific communication, as it was my first time writing a research paper and presenting my studies in the research paper. This really helped me gain some valuable experience that, again, I will use in the future. The last takeaway from this experience that I want to share is how I have a greater understanding and interest in metabolism and biological concepts. These two, biology, metabolism, and medicine, are concepts that I love to learn and explore. So having this opportunity was incredibly valuable for me. I just want to say thank you to the Talaria and Abwis team for giving me this opportunity to do research, work with a mentor, and meet such amazing people in this program.